So this is the last session in the chapter Biotechnology Principles and Processes, where we'll try to complete the remaining part of the bioreactors, and we'll see the last step of the chapter, that is downstream processing. So we are telling bioreactors, which are also called fermenters, they are the vessels in which the raw materials are biologically converted into special proteins or special products using microorganisms. Sometimes we use plant cells, sometimes we use animal cells or human cells, correct? So what did I tell? Bioreactors are fermenters. They are vessels in which the raw materials are biologically converted into special products using microorganisms. Let us write on bioreactor. It is also called fermenter. What is it? It is a vessel in which raw materials, whatever we add, it is a raw materials. Raw materials are converted, right? We can write they are biologically converted because microorganisms are converting them. Are biologically converted into specific proteins, right? Or specific products. Now, how are you converting the raw materials into specific products? So we are using a genetically modified organism, genetically modified microbe you can use, or a plant cell, or an animal cell, or a human cell, using genetically modified, right? We are using genetically modified microbe or a plant cell or an animal cell or human cell, right? So we use these genetically modified cells and we provide the raw materials to get our desired proteins in large scales. That is called bioprocessing. We do it in a bioprocessor, right? So a bioprocessor or a bioreactor, so in order to convert the raw materials into specific products, it has to provide all the optimal conditions, right? In terms of temperature, pH, substrate concentration, microorganisms, they require salts, they require vitamins, they require oxygen. Then only they can convert the raw materials into desirable products. Means all the optimal conditions which are required for bioprocessing are provided by the bioreactor. So we can write a bioreactor provides all the optimal conditions. It provides all the optimal conditions which are required for, what are they required for? Which are required for bioprocessing. What are the things? Come on, tell me. It has to provide optimal temperature, optimal pH, it has to provide optimal substrate concentration, it has to provide optimal substrate concentration, it has to provide salts, vitamins it has to provide, oxygen it has to provide, all these things it has to provide so that the process will happen. Means the bioreactor is equipped with certain techniques so that it can help in mixing. Bioreactor is equipped with certain techniques so that it can supply sterile air, oxygen. It is equipped with certain techniques so that it can control the foam, because excess of foam generates a problem. Then it has a device to monitor the temperature and it can control the temperature. Means bioreactor has so many systems which are inside. We can write that point also. Bioreactor. What does it has? Bioreactor has, we can tell, temperature control system. One thing it has, temperature control system. To need to check the temperature, we have a thermostat. Thermostat will detect the temperature. And how will you control? It is jacketed, either will circulate steam or will circulate cold water. It has an agitation system, impeller, motor and all. That's called agitation system. It has temperature control system. It has agitation system. It has oxygen delivery system. It has foam control system, right? So it has foam control system, it has pH control system, we can write pH control system also. And it has a sampling port. Now, what is this sampling port? I think you understood. Temperature control, it is by thermostat. Thermostat will tell us the temperature, and circulating hot steam or through cold water we can control it. Agitation system is by impeller rods. Oxygen delivery system is by the tubes and sparger, right? A plate which is having tiny nozzles. Foam control system is by using anti-foaming agent, right? Or by foam breakers, blades, right? Then pH is by acid-base reservoir. pH control is done by adding acid and base, right? So then what is sampling port? Sampling port means it is a place through which we can extract the sample, and periodically we can extract the sample and we can see how is the progress of the reaction happening inside. So is there any contamination, or the rate of the progress of reaction is happening properly or not, we can see. Come on, tell me what is sampling port? From where the samples are periodically drawn out to check the progress of the reaction. Sampling port means through which the sample is periodically drawn out. Why? To check the progress of the reaction. To check the progress of the reaction or the process. Am I understood? So in the previous lecture, we discussed about the construction of a bioreactor. Here I am just summarizing what are we doing in a bioreactor. Bioreactor, which is a fermenter or vessel where we add the raw materials, and the biological organisms which are present there, like microorganism or a plant cell, animal cell or human cell, will convert into the specific products what we need. A bioreactor should provide all the optimal conditions which are required for the processing, like it has to maintain temperature, it has to maintain pH, it has to maintain the substrate concentration, salts it has to provide, vitamins it has to provide, oxygen it has to provide, and it is equipped with systems. System to control the temperature, system for mixing, system for oxygen delivery, system for controlling the foam, system for maintaining the pH, and it has a sampling port also through which we can take out the sample time to time and we can see how is the progress of the reaction happening. So when you check all these things, then the process will get completed. So means this is a process. Now the last step is called as downstream processing, where we will try to collect the product, extract the product, purify it, formulate it, pack it, market it. Okay, if you want, take a screenshot. We'll come and talk about the next topic, that is downstream processing. Right. So the last topic in this chapter in NCRT, they have given us 11.3.6, which is called as downstream processing. 11.3.6, right? Let's go. Now you understood what is bioprocessing? So after completing the biosynthesis, we need to go for downstream processing. We need to go to the last step, which is downstream processing. When will we go for downstream processing? It is carried out only after the biosynthesis is completed in the bioreactor, right? Then downstream processing means, what do you think? What is it? Is it the recovery of the product? Yes, it is the recovery of the product from the fully grown, genetically modified cell. We will do it after completing the bioprocessing. And what do we do here? We'll try to recover our product from a fully grown genetically modified cell. Now, downstream processing is not a single step. It involves various levels. It involves various processes. Like first we need to filter it. Then we need to separate it. Then we need to purify it. Afterwards, we need to formulate it, means preservatives, addition of preservatives to which concentration. We need to formulate it. And then after preparing everything, so we need to check it also, right? Quality control should also be done. All these steps one by one will come under downstream processing. So means it's not a single step process. It is a multi-step process, involves various chemical techniques which are meant for filtration, which are meant for separation, purification, formulation, packaging, right? Let us write on. It is carried out after the completion of the biosynthetic stage. Downstream processing is carried out after the completion of biosynthetic stage. Where did you do the biosynthetic stage? In the bioreactor. After everything gets completed in the bioreactor, then we will come to downstream processing. What is downstream processing? It is a recovery of the product. So, downstream processing, I'm using a quote, downstream processing is the recovery of the product, right? It is the recovery of the product. From where? From fully grown genetically modified organism. From fully grown genetically modified organism, isn't it? Then downstream processing is not a single step process. So downstream processing is subjected through a series of processes, right? So downstream processing is not a single step process. It is not a single step process, but it proceeds through various processes. Like, what are the processes? It proceeds through various processes like, first I told, filter it and collect the filtrate and solids. First one is filtration. After that, we'll go for separation, density gradient centrifugation, gel electrophoresis. First, we will filter and separate the liquid part separate, solid part separate. Then we will go for separation. After separation, we'll go for purification of the product. After purification of the product, we'll go for formulation. How much chemical we need to add it so that the shelf life of the product will be maintained for 6 months, 1 year, 2 years. What is that called? Formulation. Okay. After formulation, we'll go for quality control testing, right? QC testing. After QC testing, we'll check it for clinical trials. In animals, we'll go for clinical trials. After QC gives a certificate that yeah, it has passed all the QC tests. After completing the clinical trials, then we will pack it. Packaging and marketing. Of course, marketing doesn't come under downstream processing. So downstream processing is not a single step process. It involves various stages. It involves various processes. Like first one is filtration, purification, separation, formulation, quality control testing, clinical trials and packaging. Understood? Now, we can tell that if we see a biotechnological company, how it will be the setup of the company. First, they have an R&D division. First, they have a division called R&D division. So, they will prepare the genetically modified organism. So, after this, their formula works out, then it goes to the bioprocessing division. What is this called? Bioprocessing division. R&D people will develop the formula, will develop a genetically modified organism, and then it goes to the bioprocessing division where we will do the biosynthesis. After synthesizing it, then it goes to downstream processing. Then it goes to downstream processing. In downstream processing, what they will do, they'll filter it, they'll separate it, they'll purify it, right, and they will formulate it. So after checking everything, after taking it out, purifying it, formulating it, then they will pass it on to the QC lab. To whom they'll pass? They'll pass it on to another division. What is that division called? Quality Control Division. QC stands for Quality Control Lab. They'll pass it to the Quality Control Lab. In Quality Control Lab, so they will check the quality of the product. So whether it is safe, are there any side effects and all. So that they will check. After the Quality Control Lab has given us the assurance, yeah, it came out well, then it is sent into animal husbandry. It is sent into animal husbandry where we will do clinical trials. On experimental animals. Now, we need to check every product in animals, right? That's called clinical trials. We need to check whether it is working or not. So, they'll do clinical trials in the animals. After the product crosses gets the certificate from the quality control, gets the certificate through the clinical trials, then only it will come to the packaging unit. They will pack it and then it will go to marketing. So these are the vital stages. First, Research and Development Division will genetically manipulate the organism. After manipulating the organism, they'll send it to the Bioprocessing Division. We know stirrer tank bioreactor, Sparger bioreactor, batch cultivation, continuous culture. So you'll decide and you'll do it. After doing it, so whatever they have collected it, so they'll give it to the downstream processing unit. In downstream processing unit, so they will do all these steps: filtration, separation, purification, formulation. They'll purify it, and finally they'll give the product to another lab called Quality Control Lab, where they will check the purity of the product. And they will give the assurance, yeah, it came out, the product came out very well. Then we are going to check it once on the living organisms. So we'll go to the animal husbandry, we will inject it to the animals and we'll see whether it is responding well or not. For example, if you have produced a vaccine, Rdna vaccine if you have produced, so first we will vaccinate the animals. After few days, we will inject the antigen also into their body. If they are vaccinated and if you give antigen, then they should not get the disease, right? Because you are injecting antibodies into their body. So we'll see the clinical trials. After crossing the clinical trials only, then if everything passes here, it will come to the packaging and then it will go for marketing. Understood? So then which preservatives to be added also, it depends. So what is that called? Formulation. What do you mean by formulation? Formulation means addition of preservatives. What level of preservatives? Grade 1, grade 2, grade 3. Which level of preservatives to be added and to which concentration to be added? That we need to decide. And again, the purity levels. The purity levels will vary from product to product. What do you mean by the purity levels vary from product to product? The same antibiotic, if you are using it for veterinary purpose, for cow, buffalo or sheep, then it may not be 100% pure, right? But if the same antibiotic, if you are using for the human purpose, it need to be 100% pure. It need like 0.1 impurity also, it causes different problems in the human body. So that's what we are telling. The purity level of the product varies from product to product. Is it a product for a veterinary purpose? It's a product for human purpose. Depending on that, they purify the product. And again, Formulation. Formulation means addition of preservatives. Which type of preservatives to which concentration you have to add, you need to check it. And after everything goes, then finally on the packaging unit, they'll give the batch number, right? So when you take any medicine, on the medicine 5-digit batch number will be there. So each batch number they will enter in their register, and the people will sign, we checked it on these many animals and it came well, the quality is this much. They maintain that record. They maintain that record. A biotechnological company has to maintain the record for each and every batch. Who did it? When they did it? How did they check it? What are the quality control levels? They have to check it. Suppose if anything goes wrong, if anyone claims, this company should have a thing like, yeah, we did it, it came out well. I don't know whether they have taken the same company medicine or not. So this is very important step. Downstream processing is very important step, because we cannot play with the lives of people. Let it be a animal or let it be a man. So all this is about downstream processing. In downstream processing, quality control plays an important role, clinical trials plays an important role, and this is the order of downstream processing. So children, with this we finished the chapter. Now when you come to the Ncrt exercise questions, in the first question in the Ncrt exercise, they have given us a question like find out any 10 recombinant proteins and identify their uses in therapeutical values. Identify their medicinal uses, they asked. We will do that also in this class, and then we'll stop. If you want, you can take a screenshot, then we will discuss about the 10 recombinant proteins. Yeah, we finished the chapter successfully. Now let's go and see what are the 10 recombinant proteins. Okay, so any comments, you can post it in the comment box. Hope the lectures are informative, will help you to crack the NEET, get a prestigious score. Now let's work out 10 recombinant proteins which are used in therapeutic, which are used in medicines. They asked us to list 10 recombinant proteins which have medicinal value. Which have medicinal value. This we need to talk about. 10 recombinant proteins which have medicinal value. Now, I think you know, or in the next chapter, we will see there is a protein, there is a hormone called humulin. There is a hormone called humulin. So what is humulin? Human insulin produced by Rdna technology is named as humulin. What is humulin? It is human insulin hormone produced through, how did we produce? Produced through Rdna technology, children, recombinant DNA technology. So we call it as humulin. Now we know that in our society, so many people are suffering from sugar-beetle, diabetes mellitus. So it is because of the deficiency of this human insulin hormone. So for treatment, earlier when biotechnology was not there, so they used to take insulin from slaughterhouses, pork insulin, beef insulin, which is not human specific insulin. It will give some side effects. But with the help of biotechnology, we were able to manufacture human insulin only, which doesn't have any side effect. It's our insulin, right? So it's not chemical also. It is our biological product only. So thanks to biotechnology, which has prepared humulin, which is called human insulin produced by Rdna technology, which is successfully used in treatment of diabetes mellitus. Where do we use it? It is used in treatment of diabetes mellitus, right? Now, second one, if we have to see, human growth hormone can also be manufactured by Rdna technology. What is the second hormone? Human growth hormone, which is also a protein. Human growth hormone. This human growth hormone, so it can help in removing dwarfism. It will bring out to the normal growth. So it helps in treatment. It helps in treatment to remove dwarfism, right? So Another hormone, if we see, calcitonin. Now, calcitonin deficiency, what does it causes? Calcitonin deficiency causes rickets. Bones become weak. So, calcitonin hormone can also be produced by biotechnology, Rdna technology, which is used in treatment of rickets. So, calcitonin is used in treatment of rickets, right? Then, next hormone, if we go, it is chorionic gonadotrophin. Another hormone is chorionic gonadotrophin. HCG is a hormone which helps in pregnancy, right? HCG is a hormone only seen in pregnant conditions. Now, HCG, it helps in treatment of infertility issues. So, it will help in fertility. So, HCG can also be produced by Rdna technology route. It helps in treatment of infertility. It helps in treatment of what? Infertility. And another all hormones only we are listing. Erythropoietin is another hormone. So, the JG cells of the kidney produce a hormone called erythropoietin. Now, in kidney failure conditions, when the kidneys are not working, then JG cells also will not work. Then erythropoietin hormone will not be produced. As a result, the people will be anemic. So, to treat anemia or to treat the people who are undergoing dialysis, erythropoietin can be prepared by biotechnology and we can supply them. So, erythropoietin is used in treatment of anemia. It is used in treatment of anemia. And what did I tell? Dialysis. What is dialysis? When the kidneys fail, then they go to dialysis unit and there they remove the excess of ammonia and urea from the body. So, then in dialysis patients also kidney is not working. Erythropoietin will not be made. So, then they have to depend on this erythropoietin only. So far I think we listed 5. What are they? Human insulin used in diabetes treatment. Second one, growth hormone used to treat dwarfism. Calcitonin used to treat rickets. Human chorionic gonadotrophin used to treat infertility issues. Erythropoietin helps in treating anemia and dialysis. Next we can list down blood clotting factors. Blood clotting factors. Now, if these factors, they are also proteins. If these factors are not there, it leads to hemophilia. Blood will not clot, right? It might even lead to death also. Blood clotting factors are also produced biotechnologically. Like specifically, blood clotting factor 8 causes hemophilia A. Blood clotting factor 9 causes hemophilia B. So, we can produce these blood clotting factors and it will be used in treatment of hemophilia, right? It is used in treatment of hemophilia A and B. Hemophilia A and B. C is also there, but these two are prevalent. So, this also we can tell. Now, next, what can we write as interleukins? Can also be produced by biotechnology. Interleukins, we call them as IL. Interleukins are produced by biotechnology, Rdna technology, which will help to enhance the immune system. If our immune system is enhanced, we will not suffer from diseases and illnesses very soon, right? So, interleukin help to enhance immune system. What is immune system? If it is strong, we can fight, we can give a tough fight to pathogens. Next one, interferons are of 3 types, you know, alpha interferon, beta interferon, gamma interferons. We call them as INF. Interleukins are called IL, interferons are called INF. Interferons, which are produced by Rdna technology, they can be used to treat viral infections. They can be used in cancer treatment also to help the person to recover from cancer. Interferons are used. So, if normal interferons, if we give like chemicals and all, so chemicals will unnecessarily damage the body again. If you give the biological product like interferon, so it will not cause that harm, right? So, interferons are of 3 types, alpha, beta, gamma. We can produce through Rdna technology. They help in treatment of viral diseases and what I am telling, in treatment of cancer also. In the treatment of cancer also we can use them. Then Rdna vaccines, right? Do you know any vaccine? What's the first Rdna vaccine which is manufactured in India? In exercise questions, in Ncrt exemplar questions again, what is the first Rdna vaccine to be manufactured in India? It is hepatitis B vaccine. What is that vaccine? Hepatitis B vaccine. So the credit goes to the first Indian biotechnological company. It is Shanta Biotech. Shanta Biotech has manufactured this hepatitis B vaccine. Now, what we are using, this corona vaccine, is also a product of Rdna vaccine. That also we can tell, right? The corona vaccine we are taking is also an Rdna vaccine only, which is a third generation vaccine. And again, we can tell malarial vaccine is also there. Malarial vaccine is there. Measles vaccine is there. All these are the vaccines which are produced by biotechnology, which are safer than the earlier traditional vaccines. And then we can talk about platelet growth factors. Platelet growth factors. Now, what will the platelets help in? They help in blood clotting, right? So platelet growth factors, if we inject, it helps in quick wound healing. It is used in wound healing. And if we have to list one more, we can talk about monoclonal antibodies. We call them as MAB, monoclonal antibodies. So what are monoclonal antibody? An antibody which fights only against a particular antigen. So we have kits to detect typhoid, to detect corona, to detect pregnancy. So in these kits, so how are you checking? So means you are preparing an antibody or an antigen which can bind with only one specific antigen. Antigen antibody reaction will occur and the line of precipitation will appear, right? So blood grouping, in blood grouping also, what do? We take a glass slide, we divide into 3 parts. So from the kit, you add 3 drops of the reagent and you'll prick your finger. You'll add 1 drop of blood here, 1 drop of blood here, 1 drop of blood here, right? So here you added antibody A, antibody B, antibody RH. Means if my blood group is RH, AB positive, then I have antigen A. It will react with antibody A. I have antigen B also. It will react with antibody B. I have RH antigen also. It will react with RH antibody. So it will clump. So means where are monoclonal antibodies used? Monoclonal antibodies are used in manufacturing diagnostic kits. They are used in manufacturing diagnostic kits. They are used in blood grouping kits also. They used to manufacture. They are used in identification of blood grouping also. What are monoclonal antibodies? Monoclonal antibodies are the ones which can specifically interact or which can specifically help us to identify an antigen and helps us to find out whether a person is having that antigen in the body or not. So they asked us 10, so we can list down so many. I listed 11, I think. 10 recombinant proteins which are having medicinal value are human insulin, used in treatment of diabetes, human growth hormone, treatment of dwarfism, calcitonin, treatment of rickets, human chorionic gonadotropin, treatment of infertility, erythropoietin, treatment of anemia, blood clotting factors, treatment of hemophilia, interleukins will enhance the immune system, interferons, cancer treatment. Rdna vaccines are safer than the traditional vaccines. Platelet growth factors will help in wound healing. Monoclonal antibodies are used to manufacture diagnostic kits, are used to blood group identification. With this, we successfully completed the chapter Biotechnology Principles and Process. In the next lecture, I will come out with a new chapter. If you like the lectures, like, share and subscribe to my channel. Thank you.